So my name is Claudine Bruck. I'm the CEO of Prolifagen, a company that aims to prevent heart failure by treating heart attack patients with a microRNA molecule. There are about 600,000 survival heart attacks in the US every year. And my slides do not progress. I am sorry.
Brendan, can you bring up the slide deck?
Yeah.
Sorry for that.
No worries.
I think you'll have to stop sharing your screen so Brendan can share his.
There we go.
All right, I will share my screen now and start your timer, okay? Can you see okay?
I can.
Looks good.
Thank you. Thank you.
Go ahead.
That's good. Yeah. Thank you. So my name is Claudine Bruck. I'm the CEO of Prolifagen, a company that aims to prevent heart failure by treating heart attack patients with a microRNA molecule. There are about, next slide please, 600,000 survival heart attacks in the US, 20% of which will lead to heart failure. Next slide, please. These 20% represent our initial target patient population, a total of 400,000 patients in the US and Europe combined. No, the preceding slide, please. When an area of the ventricular air wall is damaged, the heart remodels during the weeks, months, and years after the heart attack, becomes enlarged and loses its pumping capacity. As a consequence of being unable to regenerate its damaged heart, more than 3 million in the US live with heart failure resulting from a previous heart attack at a yearly cost to society of more than 15 billion. Next slide, please. Prolifagen's treatment, Pro302, can prevent this devastating condition by directing the growth of resident surviving cardiac muscle cells. Pro302 is composed of a microRNA molecule and a hydrogel formulation that protects and delivers the microRNA over 7 to 10 days. Pro302 is injected into the border zone of the affected area, where resident surviving cardiac muscle cells proliferate and reseed the injury. This represents a significant step forward relative to stem cell treatment, where cells do not efficiently engraft in the heart, and those treatments have failed so far. On the right, you can see the impact of MIR302 on mouse heart. The upper heart suffered a ligation-induced heart attack. The lower heart too, but was treated with MIR302. And it is apparent that treatment has a strong effect on the lesion size at 1 month after treatment. Please, next slide. This anatomic effect also translates in a strong benefit on function at 4 weeks after infarct. Next slide, please. Before we proceed to clinical development, we need to demonstrate effectiveness in a larger heart using an established pig model. We're using a small business innovation research grant to prepare for a controlled trial in pigs, aiming to determine the extent of clinical benefit and serve as a basis for clinical development. We need 1.2 million in seed funding for that study, and then we'll be able to raise 15 million in Series A funding to complete IND-enabling studies, followed by phase 1/2 clinical study. We expect clinical results in 2024. That will be our exit point. Next slide. Our management team is composed of myself, a pharmaceutical executive with end-to-end drug development expertise, and Professors Ed Morrissey and Jason Burdick. Ed Morrissey and Jason are professors at the University of Pennsylvania and internationally renowned experts in their respective fields of cardiac development and regeneration and hydrogel delivery of medicines. Next slide, please. This is our intellectual property. microRNA induction of cardiac regeneration was granted this year in the US and Europe. And with that, I'd like to thank you. Next slide, please. And leave you with these messages to look at. Thanks very much for your attention.